And we're going to exercise out to the coffee break, but not until we hear from Marie DeMarco. And we're going back to the science section now. We're going to talk about measuring some of the fluids or some of the biomarkers, really, in terms of making a diagnosis or even some of the interventions that can happen. Dr. DeMarco.
Thank you, Lynn. Good morning, everybody. Good morning. I think I have to start by thanking Teresa for getting the blood flowing and getting our cognition up a notch. So I am going to sort of switch gears a little bit from therapeutics and interventions and talk about developing diagnostics for Alzheimer's disease. But I hope by the end of the talk you'll see how these things are all really closely connected. Before I get too far into the talk, I want to acknowledge a few key people. So the diagnostic testing I'm talking about today was done in close collaboration with a colleague of mine, Grace van der Gugten at St. Paul's Hospital. I also want to point out Roy and Swami, who will be here today and who will be standing by our poster. If you want to get more information during the coffee break, feel free to track one of them down. I'm going to talk about three basic things. So one is, why do we need better testing? The second is, how are new tests developed? What's the process? How long does it take? And the third is, hopefully I can convince you of a success story of us navigating through this process. So to the first question, why do we need better tests for Alzheimer's disease? And this really this question comes from my grandmother. We were having a conversation about what I do on a daily basis. And I'm paraphrasing her a bit here, but she really wanted to know why we need better tests. And somewhat jokingly, when she figured out what I did for a living, was wondering, well, instead of developing diagnostic tests, why don't you spend time developing new drugs? That's really what we need, right? It's new drugs. And so we had a bit of a conversation about it, and it was actually really helpful because I ended up getting this question quite a bit. Even from clinicians who are reviewing my grants. Why do we need better tests? So let's talk about that. Well, as you saw, and Hakan certainly gave a great introduction to this in the opening remarks, better tests are going to help us with earlier and more accurate diagnosis. Earlier meaning in the very early stages of the disease, maybe when you get the first symptoms that something might not be quite right. And hopefully in the future, even before you have symptoms, we'll be able to identify whether you're likely to have Alzheimer's disease. I know from working with many of the clinicians in the clinic here at UBC that diagnosis is really central to providing efficient and effective healthcare services and community services. The Alzheimer's Society is here, and they'll speak to that today, and many of you might be aware of their First Link program. There's also many associations out there that have wonderful resources for patients and families. Ultimately, we want to empower individuals. We want to give them that option if that's something they want. And we had the survey earlier in the opening session about who might want a diagnostic test if it's available. So we want to make sure we can give that option to people. Okay, so we have the benefit of early and more accurate diagnosis. We also have a better chance at stopping the disease. So unfortunately, neurodegenerative disorders, Alzheimer's, Parkinson's disease, dementia, Lewy body dementia, vascular dementia, they're all related to ongoing damage that's going on in the brain. We really want to intervene at the earliest stage as possible in order to stop much of the damage. So I give the analogy of, cooking dinner and you accidentally light your dinner on fire. You see it flash and then you quickly grab a towel and put it out. Now, if you can do that quickly, then you haven't done too much damage to the kitchen, right? There's a little smoke, but hopefully all you have to do is throw out the dinner. We want to have that same approach to Alzheimer's, that if we can tackle that really early, if we can test some of these new drugs you heard about in development and therapeutics, that we want to intervene at the earliest possible stage. Okay, so getting to the question my grandmother was really asking about is, how is what I'm doing related to the development of new drugs? So we've seen some scenarios already about therapeutics in development where there is something that shows promise for maybe stopping or slowing the disease. It's there's a lot of research that goes on in animal models. We see that it's safe to use in patients, and then we test it out. We want to test it out and see how it influences the disease. So how do we do that? Well, we enroll a large number of individuals with the disease. So if we have a therapeutic targeting Alzheimer's, we want to enroll a large number of people with Alzheimer's or that are at risk for developing Alzheimer's. And this is based on the diagnosis given by a doctor. So a doctor takes all this information they have, the symptoms, maybe some simple imaging tests like an MRI, and some cognitive assessments like taking the memory tests. And they assemble all this information, and they try and decide if this person is likely to have Alzheimer's disease. Now, earlier in the disease course, this is really challenging because you don't quite have the full picture, and so they're doing their best to try and piece this together. Unfortunately, we have learned by looking back at some therapeutic trials that have failed that a large number of people, as shown and is demonstrated here by those in red, actually were found later on not to have the disease that we were trying to study. And you can imagine what that does for the therapeutic trial when you're testing a drug in someone who doesn't even have the disease. So that's not ideal. But there is a silver lining here, in that we've learned the lesson that all future trials should include a measure of a biomarker of the underlying disease process to help us make sure that we're capturing the right population. So you've heard this word biomarker being thrown around. So what's a biomarker? Well, biomarker is simply a molecule that's somewhere in our body. It could be a protein, like Neil talked about. It could be a sugar, like David talked about. And that somehow is a measure of either health or disease. So to give you an example you might be familiar with, let's take diabetes. So if you're a diabetic, you monitor your blood glucose. So you measure the amount of glucose in your blood. And so the biomarker here is glucose, because it's telling you if it's really high, you don't have good control of your disease. And so we can take that idea forward when we start thinking about Alzheimer's disease. What makes a good biomarker for Alzheimer's disease? Well, we would really like something that measures the underlying disease process, that somehow gives us a snapshot of what's going on. We know that most of the damage is happening to the brain. Unfortunately, we can't easily access the brain as far as a measure. But we can access the fluid that surrounds the brain and comes in close contact with the brain, and that's cerebral spinal fluid. Well, we'd love to do a simpler type of collection, like taking blood in the case of diabetes. That's a step further away from the brain. So we're losing more and more signal as we get further from the brain. So at present, tackling cerebral spinal fluid, doing a lumbar puncture is the best window we have into what's going on in the brain. And so we have maybe a biomarker we'd like to use, but we also need a good tool. We need the tool to measure that biomarker that's appropriate for use in patient care. So I'm going to talk a little bit about how biomarkers are developed, and then we'll circle back to a new diagnostic test for Alzheimer's. But I just want to walk you through the processes. I think it's kind of helpful for, as you are reading and learning about new diagnostics for Alzheimer's. So in this process of developing a new biofluid test, I think of it as several mountains you have to climb to get there, to get into something that can be used in patient care. And the first exciting step is the discovery, right? You have a new molecule that looks very promising for diagnosing Alzheimer's disease, and you might test it in a small number of people. So maybe I test it in this first row of people down here and see if it can tell me if they have Alzheimer's or not. So that's a great and exciting first step. If it looks promising, then what we do is we recruit more people. So now I recruit everybody in the audience and see if it works in this group, in this larger group. And I might say, well, that's great, looks like it's promising. I'm going to recruit auditoriums across Canada and see if it works in those groups. And if that shows promise, then we go on to developing a test for patient care. I put a little exclamation part beside discovery to remind me that this is where most of the media attention comes, when we have this first exciting discovery, but not always some of these later stages. So keep that in mind when you're you see news stories about diagnostics, they're often referring to this very first stage, and you might have to follow them for a couple of years until they reach these later stages. So while we're, want to climb these mountains one after another, in reality, they're actually a little bit farther apart. And that's due to the fact that they're in somewhat different worlds. On one side, we have what can be done in a research lab, such as here at UBC. And on the other side, we have steps that need to take place in a hospital, or otherwise known as a clinical lab, where we have the regulations in place, the documentation in place to follow the rigorous standards required and certainly necessary to ensure patient safety. So that leaves us, unfortunately, with a little bit of a gap where we have these exciting discoveries that are tested, they look promising, and then what happens? There's a significant lag between moving these biomarkers into patient care. And this is really where my lab fits in, where we're trying to pull these high-value targets out of the research sphere and make sure that we can get them into the hands of doctors who want to use them and patients who want the information for making healthcare decisions. So I want to end with a success story in developing and navigating this process, making it over the mountains and towards a test for Alzheimer's disease. So we heard many of our speakers talk about amyloid beta, this peptide that we know aggregates and forms clumps in the brain. We also know now that measuring that peptide in the cerebral spinal fluid is a really good marker for what is going on in the underlying pathology in the brain. So this is a thing that's providing us a window into the damage that's happening in the brain. And we also know that, as Håkan pointed out and some of the speakers pointed out, that this change happens not when you first have symptoms, but decades before you have these symptoms. So this gives us the hope of maybe providing early diagnoses. So when we looked at how we could move this test into patient care, unfortunately, the type and quality of the tests that were available weren't really suitable for clinical use. So we were stuck in this valley between these clinical world, the research world and the clinical world and the hospital laboratory. So we needed to somehow climb this last mountain. And that's what we did. We developed a test for amyloid beta in the hospital lab at St. Paul's Hospital, following all the necessary regulations. We used a modern technique that allowed us to make improvements on what was being done on the research side. So we used mass spectrometry, which is a really, really fancy scale and way to measure molecules. We have now tested this in hundreds of patient specimens, and it's used for diagnosing Alzheimer's disease in specific clinical circumstances, such as an atypical presentation, maybe someone younger in their 40s or 50s who doesn't have a strong family history. And also with the new technology we use to measure it, we're able to actually extract a little extra information than what was available previously. So we can also identify those who have a familial mutation in this region of the biomarker we're measuring, as shown here. So in that way, we're able to get a little extra information. So as far as where we are, we've definitely passed. There's a lot of information on the discovery. This happened decades ago of this marker. There's numerous large-scale studies of how this works for diagnostics, and really we're marching up this last hill towards using it in patient care. And I wouldn't say we're quite at the summit. The test is developed, but we want to next look at how this would play out in our healthcare system in British Columbia. So we want to see what the effect is on patients. We want to see the effect on how doctors make decisions based on this testing, and we want to see how it affects our healthcare resources here in British Columbia. So we're really excited to be moving forward with this particular project. So hopefully I've answered the question of why do we need better diagnostic tests, certainly to empower individuals and families if they want that information, to improve timely access to community and healthcare services. And going back to my grandmother, we really are contributing to the effort to accelerate new diagnostics, as such tools can be used in enrollment, getting the right populations for monitoring a new drug or a new lifestyle intervention. And while I focus solely on Alzheimer's disease, with the success of this particular test, we've been able to bring on a few other partners, and now we're exploring a few other diseases like frontotemporal dementia. And so I hope in the future I'll be able to tell you a few more success stories. And with that, I would really like to thank you for your attention.
Thank you very much, Dr. DeMarco. What a beautiful presentation.